So thank you very much, Elena, for the introduction. And thank you guys for allowing me for this opportunity to present 10 very formative years of my research, of my life as a researcher, actually. As part of a group of very good scientists, which was called or is called the Center for Microbial Ecology and Genomics at the University of Pretoria. And during those 10 years, I spent a lot of my time researching microbial communities in the Namib Desert. So I'm very happy to share this experience with you guys. To me, it's like looking at fun memories. So my presentation is going to focus on four different areas. But before I actually talk about science, I'm going to explain to you guys a little bit about why it's important to, why do we think that the Namib Desert is important as a biome to study. And then we'll look at the microbial ecology of the desert, the responses of some microbiomes to desiccation and water cycles in the desert, the kinetics of the microbial communities or system, and then finally, I'm going to touch on trace gas chemotrophy as an alternative means for microbial communities and desert systems like this to sort of subsist. So why do we think, and why do I personally think that the Namib Desert is such a great place to work in? Firstly, it, for me, in my opinion, is a very amazing natural lab to study desiccation gradients. Because it has a transect, a precipitation transect that goes from west to the east of the desert, as pointed out by the arrow here that you might see. And this, as I said, goes from west to east, starting with the coastline, which we normally call the subzone, where most of your water supply comes from the coast. And then you have this very small strip in between, which we call the hyper-arid zone, which is between 60 to 200 kilometers inland. And this is where you have very irregular low precipitation. And then you have what we would call the rainfall zone, where you have higher precipitation events, higher frequency of precipitation events, and then you can definitely see a striking difference in vegetation types as well, where in this zone it becomes less a desert and more like a savanna. So for anyone that has been there, you will definitely have seen this kind of gradient visually from the coast where there's really not much growing all the way to the rainfall, when you start seeing a lot of fauna and flora. So I probably don't need to highlight this to you guys, but in these kind of extreme environments, we have a lot of different environmental factors that drive not only microbial communities, but other fauna and flora to specialize. So we have obviously variations in light, temperature, nutrient availability, UV radiation, etc. Those are very important factors. But the one I'm going to focus on, and I think is more interesting for you guys, is water as a driver in the systems. And because of all these different factors, of course, you have various forms of specialized fauna and flora in the system in the Namib Desert. One of my favorite ones, if you go down here, is the Welwitschia mirabilis, which looks like a very ugly plant, but it's actually a pretty cool plant. It can live for thousands and thousands of years. I think one of the oldest is estimated to be more than 10,000 years old, although I might be wrong on that. But the two main sorts of systems I'm going to focus on here are going to be the soil microbiomes and the hypolites. And for those who don't know, hypolites are communities, microbial communities that live under the rock, as the name implies, and they're dominated by photosynthetic organisms that use the semi-translucent nature of rocks. So they live under semi-translucent rocks like parts, and they use the nature of these rocks in transmitting some of the light to be able to subsist. And as you can see in this picture, they're often characterized by this green biofilm that sticks to the rock. Before we actually talk about the science, I would be remiss not to highlight the center that supported us doing all this research, which is the Namib Research and Training Center, and more specifically, two people within the center that were pivotal for research. So these two are Gillian, as you can see on the left, who is director of the center, and then Eugene, who is the scientific director of the center. And they not only supported us scientifically, but they also allow us to, have these research expeditions, yearly research expeditions where we brought people from around the world, colleagues, friends, and collaborators from around the world, who are all motivated by similar research questions. So here in this picture, you can see some of these people, including scientists from New Zealand, Australia, China, UK, Romania, etc. And we had many of these expeditions, which are not only a way for us to network, but also to explore new research ideas. And the center itself not only supported us in terms of the fieldwork, so they provided us all the materials that we could use, and many of the materials and the cars that we could use to run our fieldwork, but they also provided us with support, scientific support for in-premises or in-house lab work, as you can see. For an example here, they have rudimentary labs where you could do some experiments, and they've been upgrading them over the years. So you can actually now, I think they now have a DNA extraction lab where you can actually extract DNA and even sequence it on site if needed using nanopore technology. But not only that, but they also provided us with great memories. I mean, for me, I will always remember the sunsets and the dunes, as well as the starry night skies. Probably the most beautiful I've seen. So now to the science. The whole research program in the Namib Desert that I was part of was really the brainchild of the scientist you can see on the left side, which is Professor Van Cohen, who's the director of the CMEG Center, the Center for Microbial Ecology and Genetics at the University of Pretoria. And he asked very simple questions. Since there is a precipitation transit or a xeric transit across Namibia deserts, there's two long-hanging questions that he can ask. One is, are the microbial communities distinct across the Namib Desert transit? And if they are, what are the drivers that shape this ecology? Now, you have to understand, when I started exploring these questions, this was 2011, and nobody was doing molecular microbial ecology in this area. So this was quite novel at the time. And the program focused on the transit, the precipitation transit, across a road which we named the C14 road. It's named the C14. So we therefore named this transit the C14 transit. And the idea of the research program was to, as you can see from the image here, the map here below, is that we would we sampled across the C14 road at specific points every year. And this sampling started, like I said, in 2011, but it kept going until 2025 was the last year we sampled. So more than 12 years actually of sampling on the same spots across the transit. And this sampling, the data research from this sampling program resulted in many publications. But one of the seminal ones that sort of is referred to as the first exploration of microbial ecology across the transit is one by Scola et al. In 2018, where we did show that not only did the communities cluster according to those xeric areas that I talked to you about. So in this graph here, which is an RDM, we have the blue points as the fog zone samples, the orange points as your hyperarid zone samples, and your green points as your rainfall samples. So not only your communities are different depending on which zone they were sampled at, but also there's other soil chemistry drivers that are driving the separation on top of the precipitation. So you have things like pH, which you expect, organic matter, which you also expect, phosphorus, but other things like all these salts and nitrates that are shaping the communities one way or the other. So this, while not being groundbreaking, sort of confirmed the hypothesis that we had that the transit was not only different in terms precipitation, but also in terms of soil chemistry and microbial community structure. What about the applets? So in my case, I'm very fascinated by applets because you can see in this picture, I find them quite striking, beautiful, very green, upside down, as you would call them, some people would call them. And to me, the striking thing about them is that some of these things could be ancient. They could be thousands of years old, because if you can imagine, they're living under those rocks, and those rocks haven't tumbled down for thousands of years. It's probably stayed there for thousands of years. So whenever I was looking into one of these communities, I thought I'm looking into prehistory. Right? So I was very curious about how these communities were shaped across the transit. If the soil communities were different, then I would expect the uplifts to also be different across the transit. And as you can see from the PCOA plot I'm showing here, both in terms of microbial composition, but also in terms of functionality, you could cluster your samples according, or your uplifts according to the three different zones that we sampled. So here you have the fog area, the upper, the game, and the rainfall. Now it's worth noting here that this is based on 16S Illumina sequencing data, and the functional capacity was predicted using PyCrust. So it is predicted capacity, not measured functionality. Okay? So this led us to the not very surprising hypothesis that the precipitation across the transect is a big driver of community structure, right? Of soil microbiome, not only soil microbiomics, but also uplift community structure. And you can not only see this through the sequencing data, but you can also see this visually. So this is what a Namib Desert pavement looks like in the Namib Desert. And this is what it looks like when there's a rain event. This is a week just after a rain event. So as you can see, the whole ecosystem changes very, very, very significantly. And this is because we believe the biological communities in the system are primed to react very, very quickly to water. And they're not dead, they're living things, obviously, that are just dormant in a state of anhydrobiosis. And as soon as water is introduced into the system, they react very quickly, and then you will suddenly have a bloom, as you can see. Now, I didn't see this one. This one was taken in the first year that they sampled. I was very lucky to see it twice during my sampling trips. One in 2019 and one in 2024, I think. And it really is striking the difference once it rains, everything just becomes green. It's amazing. And of course, in terms of what this means for microbial communities, is that, as I pointed out before, we do believe that in most cases, they are in a state of anhydrobiosis, where you do, under certain water activity, your metabolic potential is quite low. You're just maintaining your metabolic potential without much activity, and you're primed to react very quickly once you have that pulse of water. And this is a graph that was drawn by these two colleagues of mine, Jason and Gilda, who basically did a literature review search, and they drew this graph based on the data they got from the literature review. So to explore more the reactivity of soil microbial communities in the desert, two colleagues of mine, JB and Carlos Sobrano, went about to design this experiment where they measured RNA levels of expression in plots that you can see here in the foreground that were subdivided into different quadrants. And these plots were sampled under desiccated conditions or natural conditions and watered conditions. And the levels of RNA expression were compared between the two different states. I just want to point out, you can see on the background, just if you squint, you can see the station, the water tank for the station on the background. So the two questions my colleagues wanted to answer with this experiment is, what are the metabolic processes that are active during this anhydrobiotic conditions, or this anhydrobiotic state, and how does the state changes once you add water to the system? Now, to cut story short a little bit, the first thing that they found that was perhaps a little bit surprising based on our hypothesis is that they found that most of the core metabolic pathways were being expressed under desiccated conditions. And I have two of these shown here below in these diagrams. The arrows of the diagrams, the thickness of the arrows is representative of how much expression for mRNAs were found for the specific steps, right? And for instance, I just highlight these because these are two of the main processes we focus on when we study soil microbiomes. So for the nitrogen cycling process, they found that the soil nitrate was a dominant source of nitrogen, and this was driven by the glutamate synthase. And for cycling, they found things like glycerol to be the dominant carbon sources, which drive inorganic phosphate uptake. I also found a bunch of resistant genes to be significantly overexpressed, and these include chaperones, etc. Now, someone in the audience might point out, before they do it after the talk, they might point out a very pertinent question or comment, which is that we didn't do any depletion of non-active DNA or RNA in this study specifically. So the RNA you might be looking at might be actually residual RNA or legacy RNA rather than active community. And I would say that's quite a good comment, and there's a valid criticism of the study. But one thing people cannot deny is that when you have water to the system, there is a very significant change that cannot be due to just legacy RNA, but rather an active community, right? A change in the active state of your community. So as I said before, they did the same experiment with desiccated and watered plots, and in the watered plots, they sampled 10 minutes, 1 hour, 2 hours, etc., up to 3 days, or 7 days actually. Here, as you can see from the PCOA on your right, there is a big shift from the dry soils to when you add the water into the system, the community completely changes. And this is a PCOA based on the levels of ribosomal RNA in this case, not mRNA, but ribosomal RNA. So you're looking at the abundance of possible abundance of specific toxin here. So the community changes completely. Over 10 minutes, it changes even more after up to 7 hours, and then it starts reverting back to the dry state as the water as the water in the system dries up, right, and dissipates. So this is a very clear indication that obviously those communities are reacting to water addition, and they're reacting very quickly. And you can see this also by looking at the specific toxin that are being over expressed, or they're changing abundance as you add the water. So based on the data they got, they divided these toxins into early responders, middle responders, and late responders. Now, I'm not going to go through the graph, but very quickly I just want to highlight that the early responders, if you look at the taxonomic annotations, you see that many of these are your alpha proteobacteria, your Coryphaxa. These are fast, especially the alpha proteobacteria, bacteria. These are fast growing, or what you would expect to be fast growing microorganisms or bacteria. And then if you look at the late responders, interestingly, what you see is that at hour 7, you see the spike in the abundance of cytophagia, which are your microeukaryotes, but also gamma proteobacteria. And gamma proteobacteria are actually the myxobacteria. For those who know their stuff about bacteria, myxobacteria are predatory bacteria. So this led my colleagues to the model you can see here on the right, which was that in the absence of water, in their anhydrobiotic state, communities are mostly either inert, dormant, in spores, or they're expressing genes that are related to autotrophic metabolism. So things like carbon fixation, chemotrophic carboxylases, or visco, etc. And they're also expressing stress-related proteins, as you would expect. But once you have water, very quickly, within 10 minutes, the whole landscape changes. where the fast growers, including viruses, are expressing a lot of different genes, including motility genes, import and export genes, as well as reproduction genes. That basically allows them to reproduce and expand their colonies, right? And this in turn will attract a bunch of predatory microorganisms, including mixobacteria, but also your arterial predators, to come and feast on this new bacterial activity. And after 7 hours, this then progressively, as what dissipates from the system or evaporates from the system, rather, this goes back into the anhydrobiotic state. So even though we might have a background of legacy RNA to account for in this data, I think this is pretty strong evidence to indicate the microbial communities that are served in the Namib Desert react very, very quickly to change. And this gives you a very nice picture of how the dynamic goes as water through the cycles of wetting and desiccation. Now, what about apocalypse? In the case of apocalypse, again, it's much more tricky to do a similar experiment for many reasons, but one of those reasons is the fact that apocalypse are very heterogeneous communities. So, because the rock they are supported on are not the same, you don't find the same rock just represented everywhere. You will find that these communities are often very heterogeneous. They're not one and the same. And therefore, and if you think about watering these communities because they're under the rock, you have some logistic problems and limitations there as well. So instead of doing something like that, we're sort of limited with DNA and 16S, and because they're very slow growing communities, it's very difficult to do microcosm experiments. But we did try to understand how the communities or how the development of these prolific communities might be affected by differences in water availability. So to do so, we did a 7-year experiment where we established two different what we call microarrays, or not microarrays, or rock arrays, or stone arrays, whatever you want to call them. One of which you can see down here in the screen picture where everyone is smiling. And those arrays basically were composed of quartz, which was locally sourced. So this was quartz you find around the area, which was scrubbed out of any biological material and then sterilized and put back into the soil. Some quartz, which has a similar part transmission, so allows for similar levels of light to be transmitted through it. And this was cut from a rock. Then we also included, as a negative control, we included a rock which is not translucent at all, and we don't expect anything to grow in it at all. Right? And we had two of these arrays. One we call the inland array, which is in the rainfall area, and a station array, which is in the upper area. And we monitor these, we sample these using sacrificial sampling over 7 years, the triplicates, both of the rocks under the rocks and the soils under the rocks. And we did 16S and sequencing, and Illumina. And to cut story short, what we did find, which was very striking, if you look at quartz, but marble is also very similar, is that if you look at the arrays, which is in the rainfall zone, we can see this nice progression over the years of cyanobacteria. And if you recall, previously I mentioned that cyanobacteria and other photosynthetic microorganisms, we believe, are the pioneers that drive the development of these communities. And even visually, we could see this. So from 2018, we could already see visually that some green biofilm was growing under the quartz and the marbles themselves. And this, so not only do we have visual confirmation, but we also have confirmation from the DNA. But for the station quartz, we could not see any trends, and we also didn't have any visual confirmation of haplets being developed. We do have 2 years when there is slightly higher percentage of cyanobacterial compared to others, but I do believe this is a bit of an anomaly, and probably we would have to wait 10 or more years for anything to develop in this site. So this, again, is a very clear indication for us that water availability has a huge impact on the time scale of how these communities develop in the desert. And finally, I just want to briefly mention the whole concept of what we call aerotrophy or trace gas chemotrophy, which at the time when we first heard about it and we were collaborating to study it, sounded like really groundbreaking research. Nowadays, I think a lot of people are actually looking into it, so it's not as cool. It's still very cool, but it's like people might not think it's cool. But this is sort of for us was a paradigm shift from thinking about communities and deserts as being reliant on things like phototrophs that use light to generate energy. And this always causes a paradox because phototrophs need water as well, so they will be extremely reliant on water. To a system where you have trace gas chemotrophs that can use things like hydrogen and carbon monoxide from the atmosphere to produce energy. And these processes would be then driven by proteins such as high affinity type 1 hydrogenases, more specifically high affinity hydrogenases that can capture hydrogen at very low concentrations, as well as carbon monoxide dehydrogenases, which people know about. And these would then provide energy to drive the Robisco CO2 fixation, right? So this is the sort of paradigm shift that we started looking at. But when I say we, I actually mean our collaborators in Australia because the first people to actually study this seriously in the deserts was Belinda Farrer and Chris Creek, which we collaborated with. And I think one of the first reports they did was in 2017, what I show here in Nature, where they actually did microcosm experiments with soils that they took from Antarctica, and they demonstrated that these soils could oxidize trace gases. And this trace gas oxidation supported primary production in these soils, right? Since then, through our collaboration and collaboration with others, they were able to show that this happens in many different deserts, not only Antarctica, but also in Australia, and Namib, of course. And now, through many different studies, I think many different groups have shown that this process might only not be important in deserts, but also globally in other terrestrial and marine systems. So I'm just going to focus this study on the Namib, that Belinda and Dana, well, Belinda is a student of Belinda, did when they joined us, Ben in this case joined us for one of our expeditions. So as we were doing the C14 transact, she also sampled soils and she took them back to Australia and did microcosm experiments with them to measure hydrogen uptake rates in these soils. And their question was, since the communities are different across the transact, are the hydrogen uptake also different across the transact? And you expect yes. But the first thing I want to point out to you guys before I show those results is even just on the basic metabolic level, you'll see very big differences between the different points in the transit. So if you compare the fog to the upper arid to the arid, which in our case here would be the rainfall zone, you can see the differences not only in the number of 16S copies that you find, with the fog zone being very depleted compared to the other two. We also find differences in the respiratory bursts, so the respiratory capabilities, and the ATP levels, so the number of ATPs you find in the soil. Interestingly enough, I want to point out that these two first points in the transact that she sampled, the Donna sample, C14.2 and C14.4, which is 20 and 40 kilometers from the coast respectively, those two actually had levels of ATP that were bigger or are comparable to the ones you find in the middle of the transact, which that for me is quite interesting because you expect them to also be lower than the other points in the transact. Now, if you look at the hydrogen uptake itself, the really interesting results here is that not only those two sites that I just mentioned have a higher hydrogen uptake oxidation rates than many of the other points in the transact, but once you add water to the system, Those two sites in particular, as well as C14-6, had a huge increase in the rate of hydrogen oxidation. So this means that in the fog zone specifically, hydrogen oxidation seems to be a very important trophic strategy for the survival of these systems. Interestingly for me, I would expect the upper arid region to also have increased hydrogen oxidation rate, but for some reason it doesn't. This could be for a variety of particular reasons, including just the community composition. But I'm not going to go into those for this presentation. Which leads me to the last point that, okay, so these fog areas can do, the communities in these fog areas can do hydrogen oxidation at higher rates than others. What does this mean? Well, the hypothesis that Tom Cohen, the director of the center, has put forward is that this hydrogen that's being oxidized can actually be used to generate metabolic methane. A term that has been called aquagenesis, although I think this is a working term and they have to maybe try a little bit more. And what this actually means, or what the questions this poses, is that can microorganisms then be using this water that they're potentially generating to subsist or to support other metabolic potentials that we do see, or other metabolic functions that we do see being expressed in these systems, even under desiccation conditions. And with that, I thank everyone for your attention, and I'm open for questions. And I apologize for the audio.
Well, no, thank you so much for the wonderful talk. We really appreciate it. This was beautiful work to see in such a unique area of the world. We've already got our first question in the Q&A box. The question is how are you defining expressed genes with respect to desiccated conditions?
Stockman is one of the queries that the reviewers also had when they tried to publish that work, which is now a problem with are you really measuring express genes, or are you just looking at RNA that's legacy or historical RNA that during water events might have been expressed and then through the rapid desiccation just remained there, there was no degradation, right? We normally assume that RNA is very labile because there is rapid degradation of RNA, but during these conditions when you have rapid desiccation events, there might be a lot of RNA that remains. And for those who work with seeds, I'm pretty sure they've seen a lot of RNA levels there even in seeds, even if nothing is active, right? So that is one of the big questions that we still need to solve in this kind of system. And this could be solved by using technologies like as SIP, for instance, a stable isotope program, where you can actually introduce a stable isotope and then look at expressions using that, right, when you enrich for stable isotopes. But to be honest with you, despite that limitation, I still think that the evidence when you add the water, the evidence is pretty strong, very convincing to me that we are measuring, especially when water is in the system, we are measuring expression. When it's desiccated, we're probably measuring RNA that's there, but it's not necessarily being expressed.
Yeah, common challenges we kind of discuss in this area of science, right? The procedures are much different in a dry system than a hydrated system, and the complications that come with that. Thank you for that answer. So just a reminder for our audience members, you can put questions in the Q&A chat box. And so you can find the Q&A box at the bottom of your Zoom panel. It'll either be directly on the panel towards the right, or there is an option to click more, and you will see the Q&A box appear there.
Questions. For those who are interested in going, first of all, it's really beautiful, amazing place to go. One of the best times in my life were there. The people there are amazing, but please, I think that the Frank Look at the Namib Research Center are very happy to collaborate. So they're always looking for new collaborations. So feel free to contact them directly. I think they will be very responsive.
Yeah, thank you for sharing that. And while we wait for a couple more questions to come in, I will also note, so WALI, the Water and Life Interface Institute, we actually recently launched a public Slack workspace where anybody in the anhydrobiosis community can kind of easily follow up with people. Of course, all of our wonderful presenters in our seminar series would love to hear from audience members, so please also feel free to reach out to people whose work you're interested in just through email. But if you're generally interested in joining more the anhydrobiosis community, we encourage you to check out the WALI's Slack space where you can meet people and message. And all of that information will get emailed out to everybody who registered to attend this webinar. So we hope to continue these conversations, and we hope people will find new collaboration opportunities through this talk series. Well, we'll hang out for a couple more minutes to see if more questions roll in.
Maybe everyone is still very sleepy.
Perhaps we're in all different time zones, joining all around the world today. Well, while we wait to see if more questions come in, I will ask a very novice question. My background, I'm a program manager, but I had a background in some plant science, and I was just curious. In plants, we talk a lot about these time systems as we are rehydrating and kind of the conditions with which things are drying and rehydrating. And but with plants, we also have circadian rhythms. And so we've had some interesting conversations about how to contend with. There's a difference between a hot day and the change over the course of a hot day. Is that a factor also in your system, and how do you approach that?
So the closest we have to study on that is actually going back to that experiment I showed where I had a trend plot on the ground where they looked at RNA. And again, pinch of salt, you might just be looking at just the, garment RNA or whatever. But the first idea they actually had was to look at the diurnal cycle, right? So they sampled in the morning, 6 a.m., 9 a.m., midday, then 4 p.m., 8 p.m., and then midnight. It was actually pretty hectic. We had to do all of that, and we had teams to wake up. Some teams would wake up in a part, and then teams would do the other part. And we were not only measuring RNA, we were actually doing enzyme and measurements and gas exchange from the liquor, which was very primitive at the time, but it sort of worked. And what we found is that no differences at all in the diurnal cycle, which is weird. No significant differences. Obviously, there were differences, but no significant differences. So, again, pinch of salt. This could be because everything was dormant and there was no water. And when they did the water experiment, they didn't actually test for the differences. And they just tested temporal and then longitudinal study, but only on a daily, like an hourly basis until 7 hours, and then they did a daily basis, right?
Mm-hmm, mm-hmm.
It would be quite difficult to do it. I don't know how you would do it by longitudinal. If you want to water it at day and at night, maybe.
Yeah, these logistical challenges. I mean, and figuring out your time window, especially when your sampling time is so expensive or requires so many hands, that's really, really interesting. And the logistics of doing around-the-clock experiments in the field are beyond me. But sounds wonderful.
It's fun. It's very fun.
Well, we haven't seen any more audience questions come in, so I encourage you to submit them now. If you have them, and if not, of course, we encourage you all to follow up after the fact through email or through our Slack space. It's a great way to stay in touch with people. Well, I'm going to make a couple of announcements while we wait and see if we have additional questions coming in. And I will just start by thanking you, Dr. Lebre, for your wonderful presentation and sharing your work with us today. This is work that many of us haven't had the opportunity to hear much about while we've read your wonderful papers, and I really appreciate you coming and sharing with us live today. Before everyone leaves, we would really appreciate also the audience's feedback. I'm going to launch a short Zoom poll that'll appear on your screen. It should only take a minute or two to complete. And your responses are going to be helpful for us to improve our events and understand what topics and formats would be most of interest to our community. So please take a moment to fill that out, which we'll launch it right now. Okay, that should be on your screens. And additionally, while we wait and see if we have additional questions come in, I also wanted to note that this is one of the first talks in a series. We really hope to see you all at future events for the Wally and Hydrobiosis Research Tools and Techniques webinar series. As a note, the series is going to be a combination. It's a mix of research seminars like we had today, where invited speakers are sharing their research, and technical training workshops. The technical training workshops are more focused on somebody giving like a deep dive into techniques that they're experts in to kind of help our community grow in skills and connect with researchers who could advise them on techniques that they don't have access to get support on. So we hope you'll join us for that. Again, the goal of this series is to highlight both scientific insights and practical methods, and everything should be relevant to the broader anhydrobiosis or desiccation tolerant community. You'll see speakers from all different systems and all different scales. Events are scheduled through September. They generally take place once a month at 8 a.m. Pacific, and please convert that to your local time zone. But please note, the talk for next month is an hour early. It's at 7 a.m. Pacific to accommodate the time zone our speaker is joining us from. And our next speaker will be Daniela Billy. She'll be joining us from Rome and is presenting on the survival potential of a desert cyanobacteria under non-Earth conditions. So we really hope you'll join us and continue sharing this series with your colleagues and trainees who may be interested in hydrobiosis, desiccation tolerance, or anything in the world, life without water. With all that being said, once you finish your feedback survey, if you don't have any questions, please feel free to roll out. And again, we encourage you to stay connected. We'll follow up with the emails to everybody who registered for this talk today, but you can always connect with Wally on our website. You can join our mailing list, join that Anhydrobiosis community. All of these activities we do are public and are really just here to support all of the wonderful research in our field. So with that said, I will just say one more thank you, Dr. Lebre, for your wonderful presentation.
And thank you so much.
Yeah, thank you. And thank you for joining us late in your afternoon, all the way from Europe. So with that, I will wish everybody a good rest of your day. Bye-bye.
Bye.